Okay, part 7. I'm trying to keep the video parts shorter, so there may be more of them, but each one is a little bit more, is a little shorter, maybe a little bit more palatable. And we're on to slide 32, our third kind of virus, generally reverse transcribing viruses. And the kinds of viruses we're going to look at are also known as retroviruses. They have RNA packaged in their capsids, and then they go through a process that is the opposite of transcription. The reverse of transcription, which is why they're called reverse transcribed RNA viruses. Transcription is when DNA is read Transcription is DNA to RNA. Reverse transcription is RNA to DNA. Another thing that these retroviruses do is that once they've made the DNA copy of their RNA, they then stick it into the DNA of the host cell, and it just kind of rides around inside that cell. And the host cell often isn't harmed all that much by the retrovirus element, although that is not true for all cases. But the host will translate, or will transcribe and translate the viral genes as if they were the host's own genes. The process of reading RNA and making DNA is again not a process that any cell would ever do. So our reverse transcribing viruses, our retroviruses, need to encode and or bring with them a special kind of enzyme called reverse transcriptase. So retroviruses have a single-stranded positive RNA genome, but they work with that genome in a different way to how we've seen other positive sense RNA viruses. They do this reverse transcription, make DNA from RNA. They go from single-stranded RNA to single-stranded DNA, and then they rely on the cell to create the double-stranded DNA from the single-stranded DNA. And then the double-stranded DNA is integrated into the host cell chromosome. And we've actually looked at the human genome, at all 3 billion base pairs, and a large portion of the DNA in your cells is leftovers from previous viral infections throughout time. About 8%, and that's probably a low estimate, is retroviruses having got into our ancestors, having injected their DNA into our DNA, and that DNA then has just traveled through time. It may not even do anything anymore. Maybe at one time it was causing viral proteins to be generated, but if the purpose of life is to replicate DNA, then these retroviruses have been very successful because they're just hitching a ride inside our cells. Once the DNA has been integrated, it can't be eliminated. There's no mechanism that we have inside our cells to look at our chromosome and recognize which part came from a virus and which part came from a human. We can do that as humans because we can understand, we can look at the sequences and we can match them. We can say this sequence matches this virus, so this sequence must have come from a virus. but there's no innate system inside your cells that can do that kind of process. So let's take a look at retroviruses.
Retroviruses are enveloped viruses. The virion carries 2 copies of single-stranded RNA plus the enzyme reverse transcriptase. When the virus enters the cell, the single-stranded RNA and reverse transcriptase are released inside the cell. The single-stranded RNA is converted to double-stranded DNA by the enzyme reverse transcriptase. The double-stranded DNA is integrated into the host cell's chromosome as a provirus. Some cells carry the provirus in the latent state and no virions are produced, whereas other infected cells continuously synthesize new virions. The viral DNA is transcribed to produce one long polygenic messenger RNA. The messenger RNA is translated onto a long polyprotein, which is then cleaved by a viral-encoded protease to yield the individual proteins that make up the virion.
Okay, there we go. Reverse transcribing viruses, or retroviruses. There are some other kinds of reverse transcribing viruses that aren't retroviruses, but I'm going to treat them one and the same. Okay, so that's the end of our look at how different kinds of viruses go through the process of synthesis. The last thing I want to do in this set of slides is to take a look at how an infected cell knows it's infected. How do we fight back against the viruses? And this goes back to ideas that we visited when we looked at the immune system. There are lots of different kinds of cells involved in the immune system. One kind of cell is called a natural killer cell, and now we're on slide 35 there. Natural killer cells recognize host cells with foreign proteins in their membranes bound by antibodies. So here's our body cell. And our body cell has been infected with a virus. And that means there's evidence of that viral infection. The spikes that were left behind, in this case, of an enveloped virus. But there'll be other signs too, we'll look at in a second. These spikes are proteins. And we'll have various cells in the immune response that are looking for strange protein elements, bringing them to, say, a lymph node, and then presenting them to B cells being activated by T cells. And one of the primary things we're going to do is we're going to make antibodies against these spike proteins. And then our antibodies will be produced by plasma cells, and they'll go ahead and they will cover this infected cell. And here we have the constant region of our... So here's our body cell, here's our viral spike proteins, here are the antibodies we've made in response to these viral spike proteins. And this region of the antibody is the constant region. And this region acts as a red flag. It says this cell has been infected with the virus. And then our natural killer cell comes along. It has receptors that are looking out for the constant region of antibodies, and through some mechanism, through the production of some kind of toxic mechanism, the natural killer cell will cause the infected cell to die. Usually it'll deliver chemicals that cause the cell to undergo apoptosis. So that's one way. If there's suspicious stuff on the outside of cells, we can detect, and then we can't destroy the virus, however, without destroying our own cells. And that often leads to many of the symptoms, because as cells are destroyed, an immune response is mounted, cytokines get involved, all kinds of immune cells get involved, like neutrophils and macrophages, and that leads to inflammation, and inflammation then leads to many of the symptoms that we have, say from a rhinovirus that causes a cold. Many of the symptoms of drainage and swelling and fever, those actually are a result of the immune system's response. What about other viruses that don't leave behind stuff on the outside of cells? Well, we have this other mechanism. Cells, body cells throughout your body, are continually looking at the stuff that's being made inside them, grabbing those proteins inside the cell, cutting them up, and displaying them on the cell surface. So here's a protein that's been snipped up. A piece of that protein is going to be put onto this MHC molecule. The MHC molecule will be transported to the surface, and it's basically saying, have a look, I found this inside me. Is this a good thing or a bad thing? Elsewhere in the body, cytotoxic T cells have been trained to recognize bad antigens. In this case, a virus is inside the cell. A virus is making viral proteins, and pieces of the viral protein are being displayed, and this cytotoxic T cell can recognize that piece of viral protein, and it now knows that this cell has been infected. It'll then deliver, once again, chemicals that will cause this cell to undergo apoptosis. Not mentioned here, but also we have the we also have the interferon response. That's when a cell is surrounded by other cells in the tissue. This cell in the center gets infected with the virus. That causes the cell to release cytokines, interferon. That primes the cells around it to look out for signs of viral infection. And as soon as these primed cells are infected, this cell is dead, and these cells will immediately kill themselves. And the hope is then that spares all the other cells surrounding. It's like a fire break. There's a big fire. What you do is you tear down and you dig up. Say the fire is in a forest. You cut down the trees in a circle around the fire, and that means the fire can't spread outside of that circle. The same idea happening here with interferon. So slide 37, and the last one for this set of slides. Synthesis and replication. Compare and contrast DNA viruses, positive sense RNA viruses, and negative sense RNA viruses, and reverse transcribing viruses in terms of what they do, what molecules they bring with them, what molecules they make involved in replication. So our DNA virus, it doesn't need to bring anything with it to make proteins, but typically it does bring with it DNA polymerase to prevent it from having to wait around for the cell's DNA polymerase. Our RNA viruses, they require some help to make proteins, they need this molecule called replicase. Positive sense, negative sense, again, do things a little bit differently. Whether they have to bring the replicase with them, or whether they can just go ahead and make the replicase in the cell. Reverse transcribing viruses, they require this molecule called reverse transcriptase, and they do things differently. Our retroviruses, they insert their DNA genome once it's made into the host genome. We looked at the idea of antigenic drift and antigenic shift. Make sure you know the difference between those, what causes them, and the outcomes. And then we looked at a couple of ways involving the cells of the immune response. We looked at how natural killer cells work to recognize and kill infected cells, and how cytotoxic T cells work to recognize and kill infected cells. Okay, that's enough for now.